Right, okay. Well, I'd like to thank the organisers for inviting me to give this presentation. And what I'm going to actually talk about is European guidance for unwanted immunogenicity, which really is CHMP guidance, because that's the body that actually sort of coordinates all of this. I'm not going to go through guidelines because that's terribly tedious, and you can read them yourself if you really want to. So what I'm going to do is basically start off and give perhaps some reasons why there is regulatory concern about unwanted immunogenicity, then mention what guidance is available, and then perhaps give a little bit of information about how these documents have come about, and particularly which new ones are going to be around and what's going to be in them and what's not going to be in them. And then go on to just point out some things which I think are important, and this of course is a personal view, some things which I think are real issues. And then go on to perhaps finish, if I've got time, to just point out some problems with trying to set sort of across the board risk levels for particular classes of biologicals and how that can be problematical. Having said that, I might not get round to that because I've got quite a lot, and I don't, so is that clock right? No. No, damn, that's what I thought. Can't be. Yes, okay, because I unfortunately forgot to put my watch on this morning. It shows what happens when you get old and Alzheimer's sets in. Anyway, right, so first of all, I don't need to really mention much about unwanted immunogenicity, as you've had a whole day of it, and you presumably are here because you're interested and know quite a bit about it anyway. But I think it's important to realise that basically all biologicals have the potential to be immunogenic, and nearly all of them are. And this is a very incomplete list with various different biotherapeutics down here, showing that they have been immunogenic or have been reported as being immunogenic in the past. And as I said, this is a very incomplete list. It doesn't include any of the monoclonal antibodies which we know are immunogenic. But it does really just remind me to say that there is no generality about what is immunogenic and what isn't. It really seems to be just random. And more or less everything is immunogenic if you look hard enough, with just one exception, I think. Another important thing to realise is that the sort of incidence of unwanted immunogenicity also varies enormously between different products and also between different patient groups and different dosage regimes and all the rest of it. With some biotherapeutics apparently being non-immunogenic, and others really very, very immunogenic, such as from leukocytes here. And I think the only real example that I know that really stands up as being non-immunogenic is GCSF. There seems to be something about GCSF which really means that it never makes antibodies in patients. I've never seen a single example. And when you think of the use of this product, which is a high-use product, there's been millions and millions of doses of GCSF been put into patients. There's never, as far as I'm aware, ever been any real instance of immunogenicity. The other extreme, you can have products that are always immunogenic and can be immunogenic in every recipient. An example I can think of that is not in this slide, but that's GM-CSF, which is remarkably similar in size and some other properties to GCSF, but completely different in its unwanted immunogenicity. If you try hard enough, and clinicians have tried hard, if you put enough GCSF in your patients, they'll all make antibodies. So you really have these extremes. Okay, so just to sort of summarize on that, unwanted immunogenicity can range in its effects from being benign, so you have antibodies that are there but don't really seem to do anything, non-significant anyway from the clinical point of view, to serious life-threatening problems, depending on the therapeutic and all sorts of other things. The consequences are really sort of twofold. Firstly, there are consequences on efficacy, which is usually seen as reduction in the clinical response to the biotherapeutic, but there are also consequences on safety. And safety issues can occur even when there is no loss of efficacy, and vice versa. And the acute consequences of safety are things like infusion reactions, anaphylaxis, and there are also non-acute consequences, which can be very serious. So this is a serious problem, unwanted immunogenicity, or at least a potentially serious problem, and that's why there is regulatory concern about it. And probably the reason why immunogenicity is so complicated is because there is no single underlying cause. There are a whole range of, probably a plethora of different causes that can be responsible and can also interact and synergize or even sort of counteract each other. And some of these have been mentioned already. I've got a list which again is incomplete. There are certainly both patient and product-related factors important for immunogenicity. Obviously, there's the molecular structure of the product itself. Does it contain novel epitopes or other things that might be cause immunogenicity? But there's also product impurities. The formulations used can be very, very important. Whether it's got aggregates are more immunogenic normally than non-aggregates. The protein, its biological properties itself, is it an immunostimulant? GM-CSF certainly is, and that probably explains why it's so immunogenic, whereas GCSF isn't, which may explain why it isn't. Also, how it's being used, the dose, route, frequency of administration, duration of therapy, the immune status, and all sorts of other things relating to the patient or the recipient of the antibody, and also whether there's been previous exposure to the product or something like the product. And this is an incomplete list, so you can see why it's so complicated. And the real challenge of unwanted immunogenicity, at least from the regulatory point of view, is that it seems to be impossible to predict the things that you would really want to predict, such as the incidence of unwanted immunogenicity, the characteristics of the immune response, and particularly the clinical consequence and significance of such immunogenicity. Because the most common scenario is that you have unwanted immunogenicity, you have antibodies, but they don't actually do anything. And so it's very important to know what the clinical consequences are of the antibodies. And because you can't predict these, they need to be addressed in appropriate studies, normally called immunogenicity studies. So the current position, and what I mean from current position is the current regulatory position, is, as stated on this slide, I think testing for unwanted immunogenicity is integral to product development, basically at all phases, for establishing or ensuring a number of things, which include the obvious one, the clinical safety of the biotherapeutic, but also other things, product comparability. Products almost always change over their sort of life, and very often that can impact on unwanted immunogenicity. And there is also a kind of specific scenario of when biosimilar products are developed, which there's a lot of interest in at the moment, which has particular challenges from the immunogenicity point of view. Okay, so because of that, because of the regulatory concern, a number of guidance documents have been produced by a number of different regulatory authorities or related bodies, if you like. And these are generally quite similar, although there can be differences in some stress and what the guidance actually covers. The first guideline, which came out, as far as I'm aware, was the one produced by the EU, normally called the EMA guideline, which is not actually the correct name, it was actually the CHMP guideline. This came out some time ago. I think it came into force around 2008. So it's been around for some time, and it's a pretty big guideline, as guidelines go, at least the European guidelines. And that's because it's pretty much all-encompassing. It's a general guideline, and it basically covers, and this is on this slide is the basic, like the table of contents. It basically covers just about everything you would really want to know about immunogenicity and how you would assess it, at least from the regulatory point of view. But it's relatively superficial and it's general. It has no specificity. It doesn't mention any particular products, I don't think. And so that's what has been around for some time. It's got quite a lot of this kind of core general stuff, and then it's got two annexes at the end, one of which gives further information on methods, because the methodology was seen to be a particular problem by the regulatory people from submissions that they'd had. They had some pretty terrible submissions with quite awful methodology being used, and that's why there's a kind of expansion annex on this. And there's also an annex which shows you a strategy, which I'll show in a minute, which we thought, when we drafted the guideline, is particularly important to have in place right up front. Okay, so that's the guideline. As I said, I'm not going to go through it, But I thought I'd just show one slide showing the kind of thing that it contains, the kind of statements that it has, just to show you, if you haven't read it, what sort of level of information it gives. This is just from one bit of it about the immunogenicity assessment. It says immunogenicity studies need to be carefully and prospectively designed to ensure all essential procedures are in place before commencement. This includes then a whole list of things that you really have to think about. And I think this is a particularly important point, but it just shows you the kind of detail this goes into. It's not great detail, but it does give you some quite precise information, if you like. Okay, and as I said before, it stresses the need for a strategy to be in place right up front before you start doing anything. Because if you don't have the strategy in place, you'll find you've forgotten something when it's too late, and you'll have to very often go back and do everything again, which actually has happened to some companies. So this is very important. This is the strategy diagram which is in that European guideline, and it's basically just an example. You don't have to follow, you don't have to have this strategy in place. What you have to have is something like this. And it includes important things, I think, which would have to be in every strategy. How you take the samples and when you do it, how you do the screening assay, how you decide what's positive and what's negative, what you do with the positives and negatives, how you do confirmatory assays, which are almost always necessary, and then what you do with further analysis of those things you know are positive, things like determining neutralization and other characterization, and then how you put all of this together and compare it with non-antibody assays. These are assays to assess clinical markers to show whether the antibodies, if you have detected them, whether they do anything clinically. So this is the kind of thing you have to have in place. Okay, so that's the general guideline. The general guideline has been around, as I said, for some years. It's been quite widely used, and it's been generally, I'd say, well received. Some people have moaned about it, but in general, it hasn't caused a great concern by any of the people who have used it, and these are quite wide. They certainly include manufacturers and regulators themselves, but other people as well, academics, people who may be interested. But there has been one ongoing criticism that is that the guideline is too general. It doesn't contain anything that's specific about particular products. And this is not really surprising because it is a general guideline. And it was generally considered that it wouldn't be possible or even desirable to write specific immunogenicity guidelines for example, all products. It would be impossible. You'd have thousands of guidelines which would be unusable. And so this has been generally sort of not done, at least by the EU. But it has become, I think, apparent more recently that some product classes might merit more specific guidelines. And one class that really stands out is monoclonal antibodies, because there are an enormous number of monoclonal antibodies now in development. There's probably the biggest group of biotherapeutics being pursued for all sorts of different clinical indications. And some are very successful, others not. And there are immunogenicity problems with them, which in some cases are a bit different to, you might say, general biotherapeutics. And because of this, this was debated at some length first by the Biosimilars Working Party of the EMA, which basically drafts these guidance documents. And it was generally considered that we should at least think about producing a specific immunogenicity guideline for monoclonal antibodies intended for clinical use. And this was decided to go ahead, and it went through the normal EMA guideline process, which is quite complicated. It starts off with a sort of feasibility document, and that was drafted some time ago, I think 2009, and after that it was decided, following comments received on it, that we would go ahead and draft the full guideline. And this was done, and that was finally completed in May of last year. It then went out to various consultations, including a public one, which was done during last year, and that was actually completed in actually June of last year. And there were a huge number of comments. The guideline at that stage was about 8.5 pages, and we received 180 pages of comments on it. And that took some time to actually revise it in line with those. But that's now been done, been extensively revised. It looks quite different now to how it did before. And this revision has been accepted by the Guideline Consistency Group of the CHMP earlier this year. And this slide is now out of date because the guideline actually went to the CHMP for approval 3 weeks ago, and it was approved. So it is now finalized. It hasn't actually been published on the EMA website yet, because that takes a few weeks, but I assume it will come out probably next week or the week after. So it will then be in force, and you'll be able to actually locate it through their system if you can work out how to use their system. Okay, so what were the kind of comments we got from this consultation? I said this 180 pages of comments. Well, firstly, we got conflicting reviews received, although I should say the majority were supportive. It's always good getting conflicting reviews because you can basically just ignore them, because one person will say one thing and other people will say precisely the reverse. And there was lots and lots of this. And so that was something we did have to think about. There was a general comment that the new guideline should be an annex rather than a guideline by itself, because people thought it would be good to read it with the general guideline. And we had actually thought that too, but it was not possible within the legal jargon of the EMA to call it an annex. An annex means something else, which I've never actually worked out what it is, but it doesn't mean an annex like English people think of an annex. So you can't use annex, it's something legal. But we have, I think, got around that by using a different word, as I'll show in a minute. Some comments were contradictory. Some people said, for example, there should be nothing on assays at all, because assays are trivial. Everybody knows how to do assays, and you shouldn't have anything on assays because this might set things in stone. Other people said there should be much more on assays. Assays are the most important stuff. You need hundreds of pages on assays. So what we basically did was we just left it as we had in the first place. There was an enormous criticism of a section which was in this guideline, which was on prediction. And there were real venomous comments here, particularly from Big Pharma. A general comment that we got about 16 times, it had obviously been drafted by a lawyer, that basically said it was virtually criminal to have anything on prediction. All of this prediction stuff is rubbish. Should never be in any guideline. And this was quite remarkable because a lot of these comments came from Big Pharma that I know are actually doing predictive studies, because I know the people who are doing them for them. So this was, I think, a sort of a covering action. Big Pharma didn't want anything in guidelines that might say you might need to do something predictive. They want to do it, but they don't want it in a guideline. And because of that, we've actually taken it out, which I'll show how we did that in a minute, because I think we've still got it there, but it's there in a way which doesn't cause people too much problems. And another comment we got was that you shouldn't treat biosimilar monoclonals any different from any other monoclonals. And I was particularly glad about this because it was exactly what we said we were going to do in the guideline. So this was something that was very easy to address. But okay, so these were just the kind of comments that we got. There was a closed workshop on this guideline and also the guideline which was being also drafted. I'm not dealing with it here because it's not on immunogenicity. The guideline which is on biosimilar monoclonal antibodies. There was a closed workshop of this at the EMA in October last year, when lots and lots of stuff was discussed relating to both guidelines. And some modifications of the guidelines that were suggested during the discussion have been included. So what does this new guideline look like now? Well, originally, before it went out for consultation, its content was what's on this slide in the black. And what's in red is what's actually been done to it since then. So firstly, there was a section on variability in immunogenicity of monoclonals and its consequences. And this has been radically altered and basically shortened dramatically to avoid overlap with the general guideline. There was a section, it wasn't a very long section, on approaches which might be helpful in prediction. This was the thing that was absolutely decimated by the comments. And so this has been deleted, as most of what was in there really was actually covered in the general guideline anyway. There was then a section on clinical consequences, which has again been altered and shortened and combined with the risk section. There were two sections on assays, which have been changed in a kind of fairly minor way. And then there was a big section on risk at the end, which has been completely rewritten. The title altered and combined with the clinical section, and the sub-headings changed. So now the guideline looks quite differently. Also, we've added a sort of preamble that says that although it's not an annex, because that is completely forbidden by the EU system, it is actually an addendum, because the word addendum is allowed. Now, as far as I'm concerned, I speak English that my grandmother taught me. I think addendum and annex is pretty much the same, but not in the EU parlance. But addendum is allowed. So I think we've got around this problem that people originally thought that we couldn't use annex by using addendum. And I must say, Christian Schneider did a pretty good job to notice this. He first saw that there were already things out there with addendum in there so that we could use this. And so there has now been a little bit put on as a preamble saying that you do read this as an addendum with the general guideline. And the contents are now very different to how they were. Basically, the contents are basically a section on factors that affect unwanted immunogenicity of monoclonals, problems experienced with screening and confirmatory assays, a section on neutralizing assays and how they're used, and then a quite big section on clinical aspects of immunogenicity, which is really risk identification and risk assessment. So that's the new guideline, which, as I said, is actually in effect now, but should be published on the website soon. There are also some other guidelines around which are helpful from the immunogenicity point of view. One of them is the still draft, but not to be draft for much longer, guideline on basically biosimilar monoclonal antibodies, which has now been, the title's been changed to basically reflect that it now consists more or less of just non-clinical and clinical bits. And this does provide now, in its final version, quite a lot of additional guideline on comparative immunogenicity, really relating to monoclonal antibodies, but probably more general than that. So that's a useful guideline to look at. And also there's one on biosimilars containing interferon beta. And this is actually being drafted. And there's quite a lot of stuff in that on immunogenicity. And it even recommends a particular assay approach. So that's going to be quite useful, I think. Obviously, a bit limited to interferon beta, but certainly would be worth looking at if you've got interferon beta products. Okay, so what are some important points which I think are in the guidelines? I think I should say these are basically things that I think are important. Others might disagree, but I think certainly the issue of measuring neutralizing activity is pretty crucial. And also it's very important to clarify what you mean by neutralizing antibody, because that can actually differ with different products. And the requirement for neutralization assays needs to be considered. And you need to consider what kind of assay you're going to use. Should you use a classic bioassay, or should you use a competitive ligand binding assay, or something like that? In some cases, competitive ligand binding assays may be the method of choice, for example, for some monoclonal antibodies. In other instances, they may not. So you really need to think about this and not just assume that there is one common approach across the board. And I think the relevance of neutralizing antibody assessment for safety and efficacy needs to be considered. And you need to see how you would integrate this kind of data with other data, perhaps produced at other parts of the kind of assessment. Okay, and for neutralization assays need to be selected for each biological. You can't adopt a kind of generic approach like quite often you can do for things like immunoassays. And assays can use all sorts of different systems, cells or cell lines, and the cell lines can be of different types, continuously growing, factor-dependent cloned lines, or lines transfected with appropriate receptors. And you need to select a cell line which yields a functional endpoint as a response. So it needs to be quantitative. What I mean by you need to select an assay for appropriate characteristics is, I think, shown most easily by just showing you this data. This is data that we generated some time ago. Basically, these just are neutralization assays for measuring antibodies against erythropoietin. And basically what you can see here, these are all proliferation assays. So what there is up here is the proliferation, and on this axis is the dilution of antibody or anti-sera that was put into it. And there was a fixed amount of EPO used in each one of these assays. And all these assays are basically carried out in the same way. The only thing that differs is the cell line that was used. These are 3 different cell lines. And what you can see immediately is that they all work. When you put in these 2 different samples which contain neutralizing antibodies to EPO, you get a neutralization of the EPO proliferative effect. So they all work. But if you look at them, they're actually quite different. This assay cannot discriminate between the potency of these 2 samples, which is quite considerable, whereas these assays do. So if you were selecting this as a neutralizing antibody assay, you would select one of these 2 and not this. And when you look at the characteristics of these cell lines, you can see why this is the case, because this cell line has not been particularly sort of honed to respond to EPO. It responds to all sorts of different cytokines, and it's not particularly good at responding to EPO, particularly when you're trying to assess neutralizing antibodies. It's not that potent because it requires a lot more EPO, 50 times more EPO, to give these responses than these 2 cell lines, which are both very good. And the reason why these are very good is that this one is a cell line that's been grown. It's rather similar to this one, but it's actually been grown for several years in erythropoietin, so it responds very well, surprisingly. You might say, well, that's not helpful because I don't want to wait years to develop an assay. Well, you don't, because this one is probably the best approach if you do find problems with the assays, which is a cell line, actually a mouse cell line, which has the human EPO receptor transfected into it. So it responds very well, courtesy of Amgen. Okay, so what you need to do, the message of this is you do really need to think carefully about the cell lines you're going to use for neutralizing antibody assays. And it's important to do these neutralizing assays because very often they correlate with what you see clinically. Not always, but very often. And I think the best example of this is a very old one, but it's still the best one I know, which is for GM-CSF. And this was stuff that Mina Wadwa did about 15 years ago now. And basically what she did was she had patients who'd received GM-CSF. Some of them made neutralizing antibodies, and others made non-neutralizing antibodies. Even though these could be quite high titer, they weren't neutralizing. And what you can see is if you look at these patients before they're treated, and then when they don't have any antibodies at all, and then later on in the cycle when they've developed antibodies, if you look at leukocyte mobilization, you can see that with the non-neutralizing antibodies, there is no effect on the mobilization, whereas the neutralizing antibodies really do inhibit that. And basically, these patients won't respond to GM-CSF anymore, whereas these ones will, even though they have binding antibodies. Okay, so moving on, what other important points that are at least important for me? I think biosimilars are a particularly difficult issue from the immunogenicity point of view because you have to carry out comparative studies. You have to do comparative immunogenicity between the biosimilar and the innovator products. And these can be quite tricky to organize, and they can be larger than straightforward single product immunogenicity studies. So it's important to remember that if you're in the biosimilar business. Another important point was assay validation. Everybody, I think, agrees that assays need to be validated, but I think it's important to remember that assays need to be validated for their intended purposes. And you need to do validation studies to ensure all sorts of important things, some of which are listed on this slide. And you can encounter all sorts of problems when you look in validation, such as matrix effects, selection of appropriate controls can be a problem, and a particular problem is residual product in samples, and also the presence of pre-existing antibodies. But validation, very important, and as I said, for the intended purpose. And just to show an example of what I mean by the intended purpose, I'll just show this data very quickly. This is data we generated some time ago, measuring using BioCore antibodies against 2 different entirely human sequence monoclonal antibodies. And these monoclonals are exactly the same. The only difference was the CDRs. So they're very similar. And we wanted to develop this assay for a number of reasons. And we were faced with the usual problem that you don't have any positive samples when you start off because you haven't done any trials. So you don't have any positives. So you have to use what you can. And the first thing that we used to see whether the assays worked was a monoclonal antibody, a mouse monoclonal antibody which recognized human IgG. And when you put this over the BioCore machine, you get a very good response. Notice a difference in scale here. You get more or less the same response with both these antibodies, and it looks very good. So you might say, well, that's fine. But in fact, if you think about it, it doesn't really tell you very much that you really want to know, because this is just the binding of a mouse monoclonal. It's nothing like a human response. It's not even specific for these particular antibodies. So the next thing we did was try and make something a bit better. And what we did was we made rabbit serum. And again, we put these over rabbit serum against each one, and we put these over the chip. And what you can see is very low response with the pre-bleed here, and then a very good response with this antibody, and a poor response with this one. So you might say, this assay is great for this antibody, and it's rubbish for this. In fact, if you think again, they still aren't really addressing what you want, because you're not going to be measuring sort of kind of like a polyclonal response as you would induce in a rabbit. You're going to probably be measuring largely anti-idiotypic responses. So you must have an assay that detects anti-idiotypic responses very well. And we were able to test this because we had made anti-idiotypic antibodies. And it's quite remarkable what you find. This, for this antibody, which looks good from the rabbit data, is actually extremely poor at detecting anti-idiotypic responses, virtually useless. Whereas this assay, which looked quite bad from the rabbit sera data, was actually pretty good at measuring anti-idiotypic responses. So you really have to be careful when you look at validation and characteristics that you're looking at the right ones, that the ones that they are the things that you're going to use the assays for. Okay, so to sort of move on, I think it's important to realize that there is, at least so far, no perfect assay for determining the immunogenicity of therapeutics. Each assay has its own merits, etc. And you may need to, or nearly always do, need to evaluate more than one assay platform. And the assay depends on all sorts of different things. You have to select appropriate antibody controls, which can be more difficult than you think, both positive and negative controls. And then you need to do assessment studies. And very importantly, because it's a very common problem, you need to consider the possibility of interference from residual therapeutics. And basically, you can, if you think carefully, and you can find various different ways of getting around that problem, but it's very often possible to select appropriate methodology, which gives you a better response to the residual drug than others. And this, I'm not going to go through this data, but basically this just shows it's the two different types of assays, for example, a conventional bridging ELISA and an ECL-based ELISA, can have quite different responses to residual therapeutic. And that allows you to select the appropriate assay. Okay, there is also quite a lot of interest, or there has been quite a lot of interest in thinking about non-humoral immunity. Everything I've said so far has been related to humoral immunity. And people have said, well, shouldn't you be looking at cell-mediated responses? And generally, I think that is a good thing. Again, big pharma is usually extremely annoyed if you start suggesting in any kind of guidance document that you might need to do any of these assays, even though they do them. And so again, you'll find the very, very sort of cagey statements in guidelines about this. But I think, you know, really, it needs to be considered on a case-by-case basis what you need to do from this point of view. And I think if you look through the literature, you can find all sorts of examples that show that making any kind of generalizations about immunogenicity from data that you can find is very, very difficult. In other words, you can't assume because you find something with one product, with one group of patients, that you're going to find the same with a different group of patients or with a different antibody. And there's lots of examples in the literature, which I've just got a couple here on slides, where you do find differences in reported immunogenicity characteristics. In some cases, development of neutralizing antibodies in patients can clearly reduce the clinical response to monoclonals or other things. I've already shown it for GM-CSF. But there's lots of examples in literature where this is the case. For example, for Remicade, Tisabri, and Humira. But unfortunately, for other monoclonals, such as rituximab, there seems to be a much less clear correlation with that. And that makes interpretation and particularly prediction of clinical effects difficult, and generalizations dangerous. The frequency of antibody development may differ significantly in different patient groups. For example, rituximab produced, if you read the literature, 65% immunogenicity incidence, if you like, in SLE patients, but was supposedly non-immunogenic in non-Hodgkin lymphoma patients. Of course, that reflects the underlying disease of these patients. The frequency of antibody development may differ significantly in different patient groups suffering with the same clinical problem. For example, infliximab, which is anti-TNF alpha, shows a significantly higher incidence of immunogenicity in children than it does in adults. And again, I think that reflects something that's different about the disease in these different groups. And the frequency of antibody development may differ significantly in different patient groups suffering from the same clinical problem, but which differ in other ways. For example, Cimzia, the pegylated anti-TNF, induced antibodies in 12% of non-immunosuppressed patients, but only 2% of immunosuppressed patients. You might think that's obvious, but it certainly is something that is often not considered. And there may be pre-existing antibodies which can cause all sorts of problems with antibody assessment. And of course, there's the well-known example of pre-existing antibodies against gal-alpha-1,3-gal, which is present in just about all humans. But some patients actually have IgE antibodies, which can cause hypersensitivity problems if you use mouse cell-derived antibodies. Okay, so to finish off, conclude, I hope I've convinced you that immunogenicity assays occur all along the life cycle of a product, particularly when new therapeutics are developed, changes are introduced, or when a biosimilar product is proposed. And assessment requires an optimum antibody testing strategy and also validated methods and reference preparations, etc. And risk assessment may be more problematical for some products than other products because of their complexity, for example, for monoclonals. And risk may be variable even for the same antibody used differently. I'd just like to acknowledge my colleagues at MIBSC, particularly Minu Wadra, Chris Bird, Esveld Klutz, and also colleagues of the Biosimilars Working Party for the regulatory part of the talk. Okay, thank you.